The purpose of extraction is to isolate the plasmid DNA from the E. coli bacteria in order to have usable genes. A common method of DNA extraction is alkaline lysis. A kit has been developed for extraction via alkaline lysis called a Chiagen kit, which we will reference throughout the video. We begin with cells that have been transformed into bacteria. They typically are in a liquid growth medium that is removed by centrifuging and discarding the supernatant. A key in successful DNA lysis is always knowing where your plasmid DNA is located. The SDS makes the phospholipid and protein components of the cell membrane soluble. This causes the contents of the bacterial cell to be exposed to solution. Sodium hydroxide then denatures the chromosomal and plasmid DNA as well as proteins. In order to avoid irreversible denaturation of the plasmid in the alkaline conditions, acid potassium acetate is added to neutralize the solution, turning the Chiagen solution from blue to clear. Due to the high salt concentration of the solution, potassium dodecyl sulfate precipitates out, which consequently traps the denatured proteins and genomic DNA and other debris in the cell. The very small plasmid DNA is separated from the large salt detergent complexes in solution. The solution is centrifuged in order to separate the small DNA from the salt complexes. The target DNA remains suspended in the supernatant. When using a Chiagen kit, a Kia filter cartridge is used. This cartridge removes salt, detergent, and proteins from the target plasmid DNA. After being trapped in the filter, an alcohol wash ensures purity of the bound DNA. This DNA is eluted from the tip with a high salt buffer. This is then centrifuged and the pellet of DNA is washed with 70% ethanol. This wash removes any potential impurities and replaces isopropanol with ethanol, which is easily removed later. The DNA is dried and redissolved in TE buffer to protect from degradation. You now have usable plasmid DNA.